table is yours.
Okay, so hi everyone, and we will talk about Immu-vis project, deep learning for a whole slide embedding of spatial cellular data. So motivation for this project was quite simple. We just wanted to find a way for creating meaningful embeddings for tissue data. So it seems simple, but the data that we will be working with was quite unusual and quite specific. Because we use data generated by IMC method, imaging mass cytometry is an advanced method for imaging tissues. For detecting single markers, markers then are used as channels in the scans. So for detecting single markers, this method measures mass of metal isotopes in the tissue. And this method allows us to simultaneously image 40 and more markers. And the data source of the source of this data was Immu-vis. Immu-vis is an European consortium providing tumor profiling data and conducting and also supporting research done with this data. It has access to data collected from over 3,000 patients, and the consortium provided us with few hundred of such samples, which were of two types. So first there were IMC scans, and also there were cell adjacency graphs. We will talk about graphs a little bit later. But first, moving to the methods that we employed in our research and the results. Let's look at the high-level overview of the pipeline that we created for this data. So this pipeline consists basically of three encoders that are encoding the tissue IMC scan on different levels of detail. So first we have single markers of single patches, which are cropped from whole slides, whole IMC slides. These markers are encoded by first encoder, and then second encoder takes all marker embeddings for a single patch, combines them, produces embedding for whole patch, and then the third encoder combines all these patch embeddings into embedding of a whole slide. And we will describe the parts of this pipeline more in detail later in the presentation. But first thing that we wanted to do with the data, we wanted to perform some research about rotation and symmetry invariance for tissue scans. So basically we wanted to check how different architectures handle transformed data, and preferably develop our own model fitted to tissue data, to such transformed tissue data. You can see that we focused on 8 transformations, which form a symmetry group of square, which can be formally denoted as a dihedral group D4, and they are generated as follows. So we start from identity, some initial patch, then we generate 3 rotations of this patch. Then for the fifth transformation, we flip this patch upside down, and once again we apply the 3 rotations. Then we used such transformed data to perform some experiments. We cropped patches from these tissue scans, and we pass single markers through some baseline model. In that case, it was ResNet 50. Of course, ResNet 50 embeddings, ResNet 50 produces logits for 1000 classes, which are not suitable for image reconstruction, but they are still an embeddings of some sort, and they were still useful for this particular purpose, for this particular experiment. So we generated these embeddings for single markers. Then we concatenate this single marker logits into one vector. We repeat this for 8 transformation, and then we compute the threshold distances between embeddings of every 2 transformations. And the results that we got were pretty weird, because we can see that ResNet 50 and Visual Transformer for some reason perceive odd transformations as very similar to each other, but in the same time even transformations as very dissimilar. We don't know why that happens, but it kind of proves that this model does not handle such a transformed data very well. So we developed our own architecture, which was fully trained on the tissue data. It was trained on 100 by 100 patches, as well as on the whole slides. This architecture is not very sophisticated, but we aimed to create a lightweight model that in the same time would create reconstructions that are visually similar to the initial markers. Inside the encoder and decoder blocks, we use pretty standard layers. We use convolution followed by activation. For normalization, we use group norm, and also max pool for downsampling, and simple nearest neighbor interpolation for upsampling. But it worked. And also the encoder taken from this architecture after training on the tissue data was put into this pipeline as a first part of the pipeline that you have seen in one of the previous slides. Here's some example of reconstructions generated by this autoencoder. They are not that much detail, but still they they are still visually similar to the initial markers. And we also repeated the previous experiment, but on our autoencoder, or rather on the encoder part. So we encoded 2 types of patches. So these bigger patches that you can see here, and this is heat map on the right, and also we created embeddings of a small single cell patches. These were very small patches, but results are quite similar, and they are definitely more reasonable than for these 2 baseline models, but still creating a network that would be fully invariant to transformation is material for some future work. And now we proceed to the next part.
Okay, so the second part of the model takes the embeddings of the patches that were given by the first model, and since for each channel we get a different embedding, we want now to combine the embeddings and the marker to have some sort of embedding for like a spatial patch. So we have like want to take something that is that has 40 channels and want to reduce it into preferably one-dimensional representation for further analysis. So this is a little bit of a weird problem, because we don't know what channels are going to be used, and we don't know there is no predetermined sequence in those channels. So we want to somehow encode what channels are present and also like apply them to a particular channel to like associate with a particular input. And as far as I understood, there was nothing, like this concept isn't that weird, but there is nothing in the literature that applies directly to this. So our solution was to create sort of a dictionary embedding. So we take the concept of positional embedding, but instead of taking as input the position in the sequence, we just like arbitrarily select that this channel will have this representation and this channel will have this representation or other marker. And then just attach that to the input as a sort of like a guiding information. Well, so like the positional encoding works. We also want to include channels not present in the input. This is because we want the model to have some sort of a predictive power later on. We want all the channels to be outputted, even if not all of them were inputted. And the predict token is added. Predict is like, it's called CLS in the literature. It's like, it's for classifications for prediction. We use it here to get the representation of the entire input. So this is basic transformer architecture with dictionary embedding. So we just have inputs which don't have some channels, and we fill in those with mask tokens, and then we add the embedding of the channels that are associated, the markers that are associated with the channels, and we feed this into a transformer, a burst type transformer, and we get channels on the output. To get representations, we change it into an autoencoder. This is not a new idea, but like to take two transformers and have one of them be the encoder and the other one be the decoder. What's new and done by us is that we also add the marker embedding to the representation at the decoding stage to have the particular channels be outputted at the right coordinates. What's more interesting in this approach is that we can also get some correlation. We can get some correlations from how the transformer outputs the channel that is not given. So we want it to have some predictive power, and we want to harness it. We want to use it to find, to perhaps generate some data that is, to generate some channels that are not present in the input, but we would like them to be in the output to be used later. In our work, we do this by, we have our inputs be in panel 1, so there are some markers that only appear in panel 1, some that only appear in panel 2, and some that appear in both. And yeah, we can like see these correlations when we like look at the variance that is outputted by the model. We can see it gives us some insight about what model considers to be quite uncertain or certain. But this variance can be measured by a variational dropout. So we take some input, we enable a dropout in the model so that the output is not deterministic. We compute like 50 or so samples, and then we use those 50 samples to calculate the variation. We get a sort of a empirical distribution of the output, and we calculate variance out of that. So we get this plot, which is quite interesting. The color represents a channel, but each channel was fed in 10. Like each sample had all the. There were like 10 samples, and each had all the channels. And we can see that there is clustering by the batch, so like points that are close to each other are coming from the same batches. But there is also very strong clustering based on the channel, like from which channel, from which panel the channel is from, like strongly predicts where is it on this plot. And this is just a variance and loss plot. So this is quite, this fundamentally shows that the model differently understands, like sees some things are more certain and less certain and can sort of correlate them from from each other. And now the third part.
Yes, so now back to the graph part. So I was working on it, and at first we can ask ourselves, why do we even want to use graph neural networks for that problem? So it solves some issue we encountered with other models. So we can easily capture cell relations. We can get one cell and connect them to the neighboring one. It deals with the empty spaces that can be on an image. With graph, we can either add an edge or doesn't add an edge, and it will solve it by itself. It doesn't use any locality by design, so if we rotate an image, there were some issues, and if we rotate a graph, it's still the same graph. And we can easily get the whole graph embedding at the end. So how can we model the tissue as a graph? So we can either get all cells and use them as nodes and connect them to the neighboring nodes, so that would create a big graph. Or we can use those image patches and create like graph of those images, so that would create small graph, but it will be easier to train, maybe less detailed. And let's see. So I used like training pipeline called Deep Graph Infomax. We start at the top left corner. Here we start with the initial graph. It contains some initial embeddings of nodes and some adjacency matrix. We then use corrupt function to get similar graph but different. We can permute the initial node embeddings. We can add or remove some edges. We pass it through the encoder. We get two encoded graphs. And then we use a readout function to get whole slide, whole graph embedding of our true graph. And then in like discriminative fashion, we want to teach the model that all the nodes from the initial graph should have some similarity to the summary vector, and all the nodes embeddings through the corrupted graph should be as dissimilar as possible to that summary vector. So magic happens in that encoder that is getting the initial embeddings of the graph and calculates the final embeddings. I used multiple graph attention layers with Prelude, which is just a ReLU with learnable parameter. And idea behind attention in graph neural networks is when we get messages from our neighbors, some neighbors might be more important than others, so we want to scale message that they send to current node, so we can do this easily by attention. And another cool fact with attention is that we can do attention rollout and see how the edges, which edges were used the most. So the brighter the edge, the more attention it passed. This is the plot of the tissue where each node was a single cell, so we can see that this approach gives us a big graphs, so they are harder to train, especially in this discriminative fashion. And on the other plot, this is the UMAP reduced embeddings, whole tissue embeddings, whole graph embeddings, coming from the pipeline that we've described on one of the first slides. So there is also this discrepancy here. We can see the small rectangle on the top. This is data from panel 2, so there is another way to like see that the data doesn't come from the same source. And to the next steps and future work, one can manually look for interesting patterns. It's hard to get that because there is no ground truth in the biological data set. We can run the whole pipeline of our free models to get embedding of the next Immu-vis data panels. Also, we can train for reconstructing selected channels or features. Yeah, that's all. Thank you so much.